Hi, my name is Larissa Diaz. I am a doctoral candidate in coastal and marine system science at Texas A&M University, Corpus Christi. And today I'll be talking about some of my dissertation research that's also funded by Sea Grant on whether or not Texas estuaries are acidifying and what causes that if they are. So estuaries, you're probably familiar with the term, but they can be defined in a lot of ways. Here I'll define them as where rivers meet the sea, to keep it simple. So locally, we have the Nueces estuary that's composed of two bays, Nueces Bay and Corpus Christi Bay. Feeding into that estuary is the Nueces River, and laterally connecting to that estuary is another estuary, the Laguna Madre. And then those systems connect adjacently to the Gulf of Mexico, which is the sea that's local. So there's a lot of influences and dynamics in this estuary, as you can see. Estuaries are disproportionately important, though, so they support a lot of fisheries. So about 40% of the population of the United States lives within 100 kilometers of an estuary or open ocean, and we rely on these systems for food. Here locally, we have a large oyster fishery. Recreation is also important in these estuaries, so kayaking, bird watching, recreational fishing. Estuaries filter the water. So in our estuaries here, we have salt marshes that are notorious for being really good at filtering out sediments and pollution before they enter the Gulf of Mexico. Also, lands that are adjacent to estuaries are better resistant to storm surges and better buffered against weather systems than the open ocean adjacent land. So estuaries provide buffer for storms. Their role in mitigating greenhouse gases is still being investigated. I actually have another chapter in my dissertation I'm not going to talk about since I don't have time, but we're still looking into whether estuaries are more of a sink or source for CO2, and it appears to rely on which estuary it is. But some of them can mitigate greenhouse gases, such as Galveston Bay. It's looking like they can take in some of the CO2 that's a problem there, especially in that area near Houston. Estuaries are also important to organisms, so they provide habitat. A lot of times they are called nurseries by ecologists because juvenile fishes live in the estuaries and then they grow up there where it's safe and then they go out into the open ocean. Estuaries are also really productive, so they're kind of like the rainforests of the ocean. But estuaries are also disproportionately vulnerable to impacts from humans. So because they're so close to where people live and to where we need our industries to be, they can experience more severe problems than the open sea sometimes. There's less legal protection for estuaries in Texas than there is for the Gulf-facing beaches as well. And estuaries have a lower volume of water to absorb any kind of pollutant that goes into them than the open ocean. So, if you put a drop of blue food coloring in a cup of water and a drop of blue food coloring in a gallon of water, well, the cup of water will have a more blue color. It'll be a higher concentration of blue food dye than the gallon of water. And estuaries kind of work the same way with pollution. They also can't buffer against pH change as well because of their lower volume. So what happens in these estuaries to make them quote unquote acidified? Well, if the estuary takes in a lot of CO2 and that CO2 concentration increases, that makes the water's pH decrease. And this decrease in pH is called acidification, and it can cause harm to aquatic organisms. So it makes it more difficult for certain aquatic organisms, such as larval oysters, to thrive. Alkalinity is another term that a lot of my research revolves around, and I'll be discussing it today. So in a nutshell, it's a measure of the buffering capacity of the water. You can think of it kind of like an antacid for the water. And technically, it's the ability of the water to resist changes in pH. So we can titrate for alkalinity and measure these differences in hydrogen proton donors and proton acceptors. And if there's a change in pH, this kind of all ties together, right? Oyster larvae might be more vulnerable to environmental conditions and even die if the pH drops too low. So alkalinity doesn't directly influence these oyster larvae, but it does affect changes in pH and how well the water can resist that, which ultimately affects the organisms that live there. There's a lot going on in an estuary that can affect the alkalinity. This is a really crazy diagram, but what you need to know is the y-axis is alkalinity, that's TA. So anytime you see TA, that's a measure of alkalinity. And calcification is when calcium carbonate or seashells are formed by marine organisms. Dissolution is when those shells break apart. So when calcification happens, that arrow's pointing down, that means alkalinity decreases. And when dissolution happens, alkalinity increases. And when that happens, at the bottom you see that delta alkalinity to delta calcium. Well, that means that the change in alkalinity and the change in calcium always happen in a 2-to-1 ratio. So when oysters are building their shells, for example, they take in alkalinity and they take in calcium in a 2-to-1 ratio. So for part one of 2 parts of my research I'll be talking about today, I noticed from a previous study that our lab did that alkalinity and pH on average are reducing along the Texas coast. So this diagram shows alkalinity. Acidification is happening, but we're going to focus on alkalinity. And the dark green is a deeper or stronger decrease in alkalinity. So it looks kind of upside down, but stronger decreases in alkalinity are happening where it's darker. And then red is where alkalinity is actually increasing. So you might notice that, there's some variability here, but there is a trend towards a stronger decrease in alkalinity and pH in the southern estuaries. Texas is interesting because we also have this latitudinal gradient in precipitation and freshwater inflow. So the southernmost areas have less freshwater inflow and lower precipitation, and the northern areas have higher freshwater inflow and higher precipitation. So this leads to my hypothesis. I want to know the influence of river inflow on estuary and alkalinity. And Texas is a really good place to study that because of our natural gradient in this. My hypothesis is that river discharge and alkalinity have a positive correlation with alkalinity in these estuaries. The reason I think this, you probably know, but upstream, these rivers go through a lot of limestone in Texas, and our rivers tend to have very high alkalinity by the time they reach the water. So we measure that. We measure hardness, and you can taste this hardness in the water. Your appliances get backed up. You don't have to take antacids, right? But it's good for the estuary, and so I think that the more freshwater inflow here, the more higher the alkalinity of the estuaries will be. This wouldn't be true in other areas. So far, I finished with the Sabine-Neches estuary or Sabine Lake. I'm moving south, so I should have results in the other systems pretty soon. But for the Sabine-Neches estuary, I looked at the Sabine River and I looked at the Neches River that feed into this estuary. And then I looked at 3 different stations within the estuary that were TCEQ stations. Now, one kind of interesting thing about this is this mid station in Sabine Lake had a shorter time frame than the other stations had available, but I still thought it was relevant. My methods, I'm not going to go into great detail about these, but I'm always happy to talk about, you know, what I've done. So feel free to contact me if you have any questions about this. But what I really want to emphasize is that I looked at 2 different alkalinity variables in the river. I looked at alkalinity yield, which is the total amount of alkalinity, and concentration, which is the amount of alkalinity in a unit of water. And then I also looked at discharge, which is just the amount of water coming out of the river. And then I looked at the correlation between these and time and whether it was increasing or decreasing over time. And I used stats to determine this. And for the Sabine and Neches River, nothing was significant except for alkalinity concentration for the Sabine River. So the Neches River didn't have any significant trends at all, and Sabine River didn't have a significant change in discharge or total alkalinity. But the concentration was increasing over time. It does appear to oscillate on a 8 to 10 year cycle, so perhaps El Nino plays a role here. I'm going to look into that next. So yeah, it was an interesting outcome. Even with this counting that high up point in concentration that occurred in 2011, as an outlier, it still increased over time, but I'm always cautious about outliers in rivers because rivers have a lot of variability. For the estuary, there was increasing alkalinity and salinity in the inner bay and decreasing alkalinity and salinity mid-bay, so opposite trends. And you might be thinking, well, that mid-bay station had a shorter time frame, but even when I just isolated the shorter same time frame for the inner bay station, it still showed the same trend. So, you know, estuaries are dynamic and they have regions within the estuary that differ completely from other areas within the estuary. You know, some inner bay stations can have very fresh type water and outer bay stations can have very saline water, for example. So something regionally within this estuary is happening here that shows that it's not really a consistent trend. The inner bay station increasing alkalinity concentration may have to do with the increasing concentrations in the Sabine River. But you know, that mid-bay station, I don't really have an explanation for. Its correlation with salinity suggests that it might be kind of freshening, so there might be some dilution happening. But it warrants further investigation for sure. So in the future, I'm hoping to use a model to, you know, really tie these river and estuary alkalinity trends together, and then move south and look at additional rivers and estuaries and consider freshwater diversions. So perhaps, you know, I was restricted to looking at river stations that were not tidally influenced because of the mathematical models that I used, but if I were to look downstream of those, perhaps I would see something interesting. And then a large part of my research is to make everybody aware of the outcomes. So I'm going to continue to communicate these results and hopefully, you know, not just the general public, but also stakeholders like the Texas Water Development Board can be made aware of these results. So moving on, part 2 of this research that I'm talking about had to do with a phenomenon we observed in Mission Aransas estuary near Port Aransas, Texas. So in this estuary, we collect water samples pretty regularly, and we noticed that alkalinity was declining during a dry year. And also, if you looked at the picture on the left, the sediment had this black layer on top that's usually caused by iron. And when we opened it, it smelled like sulfur. So you probably smelled something similar when you cross the Laguna sometimes on a windy day, but that was the scent we got from the sediment core. So fun times, right? Smelling the mud. And if you look at this mathematically, and you look at the change in alkalinity and the change in calcium, you probably notice there's greater than a 2-to-1 ratio in these changes, so it couldn't be caused by calcification or shell building. The Mission Aransas estuary, we went to 5 stations on a regular basis, and I decided to collect cores from the 4 stations that are actually in the estuary. The southernmost station there is a ship channel station that we kind of use for comparison when we collect our water samples. So my question is, what was causing the alkalinity decline in this estuary? And my hypothesis was that oxidation of iron sulfide-rich sediments is consuming the alkalinity in Mission Aransas estuary. And this is a chemical process. It's mediated by bacteria, but it results in production of hydrogen ions, which would consume alkalinity. So those dark sediments look like iron sulfides. The stinky smell that came out of the sediments suggests sulfate or sulfur being produced. So I collected 13 cores. I incubated them for 3 to 5 weeks, collected samples, and measured many things in these samples. So I also wanted to eliminate the possibility of air pollution and calcification. And then I analyzed the data. So preliminary results suggest production of alkalinity through carbonate dissolution, followed by consumption of alkalinity by another process. I didn't collect them during a drought year. I can't control the weather. So by the time I had started collecting them, it wasn't a drought anymore. I'm still analyzing 4 of these cores, and I'm hoping that there will be some more concrete evidence to verify that iron sulfide oxidation might be the cause of this. But if not, you know, it'll probably rule it out at least. So ultimately, I really hope that my research can lead us to a better understanding of acidification in Texas estuaries and aid agencies like the Texas Water Development Board with better management of these estuaries. So on a regular basis, I communicate with and ask for feedback from these stakeholders. I hope to help make decisions regarding upstream freshwater management and diversions, help with drought management, and help to make predictions about future estuarine conditions. So after this, I'm going to create a model that will help with these predictions, and you know, you can enter a freshwater inflow event and see what the outcome might be. I also really want to let invested people know with my talks, and I'm hoping to have more talks in the future. And I'm in the process of creating a web page that will have some videos, and it will have a diagram of the biogeochemistry to help everybody kind of understand what's going on, because these are our estuaries and we need to own them and take care of them. So please keep an eye out for that web page. You can feel free to email me as well, and I can keep you updated on my email list about happenings. So thank you so much for listening. I'd like to thank my funding source, Sea Grant, the agencies that have guided me, my advisory committee, fellow members of my lab, and special thanks to my advisor, Dr. Shinping Hu, and my Sea Grant mentor, Jace Tunnell. So thank you so much for listening.